sequence of particular nucleotides. That is the promoter sequence that all of these other factors all bind onto, so RNA polymerase II can actually sit on and go, okay, let's start making the mRNA, zipping along here and making this strand from the DNA sequence. It ends up kind of lifting up and kind of coming off of there, but it's making that based on the promoter sequence and the aid from the transcription factors. So there's a promoter, and this sequence, this is crucial in forming this initiation complex made of the transcription factors and RNA pol II and all this section. It's this complex in eukaryotes responds to this promoter. There's this TATA box promoter. And this is what this promoter sequence, this TATA, or I think of ta-da, I don't know, it's not, I think of ta-da, like T-A-T-A, ta-da, this is right, boom. That little sequence right there is what is critical for forming this entire complex. It's part of the promoter sequence. That TATA box, that TATA box on the DNA is where you find upstream of all these transcriptional units or genes, right? Where to actually do this initiation complex to be able to start DNA transcription, turning DNA into mRNA. So here's a diagram that actually shows this. Here's a promoter, that's a promoter sequence. Here's the DNA, double-stranded, 5' to 3, 5' to 3, right? You have double. What happens is, in this promoter sequence, you have a TATA box. Notice you have T-A-T-A-A-A-A. And on this complementary strand is A-T-A-T. But on the main one here, we're going to go with that. Ta-da, that's the ta-da box. Okay? So several transcription factors recognize this promoter sequence. They actually are able to bind onto it, that TATA box, clamp down on it. There's multiple transcription factors typically. They bind on, and that allows RNA polymerase II to then bind onto that whole section. And this is the processive one that actually does the creating of the mRNA. This is the one that polymerizes and makes the RNA. And when it does it, it actually pops open the double-stranded DNA and copies from the 5', making new RNA transcript from the 5 to 3 direction, just like making DNA. 5', that first sugar phosphate nucleotide with the base, that's complementary to the sequence, and then it's added in the 3' direction. So it's going from 5 to 3 and adding it on. So, and notice here, if I had the complementary part, this is right at the beginning of this little green, that's the sort of the beginning of that transcriptional unit. Here's the promoter sequence, here's the beginning of the transcriptional unit, and this would be the gene or the unit that's actually transcribed and turned into mRNA. Now notice right here, this is the 5', so this is the 3'. On the parent DNA strand, that's the 3'. So again, it's complementary, so it's made in the 5 to 3 direction. Does that make sense? Right? So it's reading, it's basically detecting, okay, what is the sequence here? What is the complementary base nucleotide that we need to add to build the mRNA? And so RNA polymerase is doing kind of all the helicase, all those functions, popping it open, unwinding, opening it, and then adding the appropriate mRNA nucleotide to build that mRNA. And this ends up kind of sticking out and going, whoa, as it's being made, as it processively moves along. This is blowing that up in a bigger view, just so you can see the whole thing, okay? So we have the promoter sequence, and there's this little starting point where it actually, so we have the TATA box in this promoter sequence. This is showing RNA polymerase II, that's the one that's really doing this, making mRNA in eukaryotes. It opens up, and it starts to create that RNA transcript. And the transcriptional unit that is ultimately going to be transcribed goes from that little beginning, that starting point of that gene or transcriptional unit, all the way to the end. So this is going to zip and keep going. So this is all initiation, and I'm not showing all those little promoters. There's a lot of other promoters that bind to the promoter sequence. Transcription factors. Transcription factors that actually bind onto that sequence that allows RNA polymerase to bind on and do its thing. Now that's initiation, but in elongation, RNA polymerase II basically unwound DNA, opened up, and it's making or building this as it goes along. And it's going to continue to go along, and this is going to get longer and longer and longer as it moves, building from the 5 to 3' direction of building that mRNA transcript. So that is essentially what elongation is, right? RNA pol, unwinding, opening up, and building in those new RNA nucleotides to make the mRNA. So as RNA pol moves along the DNA, it untwists the double helix, opens it, untwists it, pops it open, 10 to 20 bases at a time, and it builds, of course, in the 5 to 3' direction. That is how nucleotides, nucleic acids, are made or polymerized, okay? Opens them up 10 or 20 at a time. So pop, unwinds, zips along, and creates the building of the mRNA nucleotides. So it progresses along at about 60 nucleotides per second in eukaryotes. How fast is that? 60 nucleotides per second, that's pretty fast. That's pretty fast. It's just very, very quick. It gets 60 in 1 second. And nucleotides making up genes or transcriptional units, these kind of segments of what's going to be turned into mRNA and then spliced together to make that protein later. A lot of these will be 100 nucleotides long or thousands of nucleotides long. There's variation depending on how big that polypeptide or that protein is ultimately going to be. So this is fast. This transcription using RNA polymerase, its ability, its enzymatic capacity is very, very fast. So a gene can be transcribed simultaneously by several RNA polymerases. Okay? So RNA polymerases, these enzymes, there's lots of them. There's not just one of each of these. There's all transcription factors that are being, transcription factors are proteins, by the way, and they can actually be made and expressed, right? So certain cells will have certain transcription factors, and other cells will have different transcription factors, and then some are general. But transcription factors are proteins, and they're part of this mRNA making process, or this gene expression process. So there's a lot of different transcription factors. There's a lot of RNA polymerase that's available inside of cells, inside of nuclei. There's a lot of this. So these basically can hop onto a transcriptional unit and simultaneously be making mRNA. Wherever, they just hop on, that the promoter sequence with the transcription factors and the RNA pol, and they zip along, zip along, zip along. So in other words, your DNA can be turned into mRNA and then protein relatively fast. You can get a lot of, within 24 hours, you can really, from your DNA, you can make a lot of mRNA that gets converted into protein. So this can be happening simultaneously. So we have that initiation, we have the elongation where it's zipping along, and then here, and actually, this is a better way to show it, it actually, I drew it coming out the top, but the way it's actually being created is it's being added to that end, right? It's going from the 5 to 3' end, so it ends up building in this direction, right? So the 5' ends up here, and it's adding on a 3', so it ends up coming out, and often it sticks out, it kind of sticks out from the RNA polymerase too. So kind of accumulating, going from 5 to 3', getting pushed back as it zips along. So it's going forward, and ultimately the transcript is kind of coming out the bottom or out the back. But this also, this elongation, new ones, after this RNA pol moves forward like this and is producing this transcript, this mRNA transcript, more RNA polymerases can sit onto that promoter sequence with the help of transcription factors and start doing the same thing, making its thing. And then it moves along, and then another one can sit on, and it moves along. So it's not like this is out of commission, it has to go from initiation to elongation to termination, and then it goes back on. These are all over the place inside of the cell, in the nucleus, so this can be happening simultaneously. In other words, you can get hundreds, thousands of transcripts of a gene in a few hours, because they can all just keep popping on and making lots of it, when it's actually needed by the cell, this particular gene or transcriptional unit. Okay, this actually shows, so here's the direction of transcription, it's adding in on the 3' end, so it actually ends up pushing this sort of down or out. And it's adding, this RNA polymerase is adding those new nucleotides onto the 3' end, right? And they already have the bases attached to them, so it's the sugar phosphate backbone with the bases. Notice there's U and there's not T, because these are RNA nucleotides, so it's adding to the 3' end. And reading the complementary strand of parent template DNA. Adding it, adding it, adding it. So this is popping open about 10 to 20 nucleotides at a time. Unwinding, unzipping, and allowing, reading it like a typewriter to create this mRNA transcript that ends up coming out. Okay, so this is elongation, in elongating or making the transcript, the mRNA. How does it terminate? The mechanisms of how transcriptional termination happens are different in eukaryotes and prokaryotes. And your book goes over this in great detail, and I don't really spend a lot of time on this. I just say in prokaryotes, the pol stops basically transcription at the end of the terminator sequence. So there's a little terminator sequence at the end, pol gets to that end, and it stops transcription. In eukaryotes, pol continues transcription after that pre-mRNA is cleaved off So the pre-mRNA basically gets cleaved off from the growing RNA chain, or the transcript, and then the pole eventually, the RNA polymerase falls off the DNA. So the mRNA gets cleaved off from that growing chain, and the pole just kind of just falls off. But basically, we just call this termination. Okay, so termination, that's it. You reach the end of the transcriptional unit, or the gene, gets there, mRNA gets cut, so now you have a free transcript, and then boom, the enzymes fall. And they can do the same thing all over again. They can go transcribe other genes and turn other genes into mRNA, okay? So we have all of this, boom, it gets to the end, termination. You get cleaving off, so you reach the end, the terminator sequence, cleaving off of that finally made transcript, the mRNA, and the RNA pole kicks off, falls off. So now it's free to be able to do this again, or go somewhere else at another location along the genome. All right, I opened with the opening slide, my title slide, and it was this picture on there, okay? And I told you this is something to do with transcription. We're gonna come back to it later. Now, what I want you to put together for a second, this is a little bit of a application question, if you will. Look at this and tell me what you think this might be, based on everything we just kind of stepped through. I will give you a hint, and I will tell you, this is DNA. This right here is double-stranded DNA going, this like tightrope right here, okay? That's all I'm gonna tell you. You're just trying to figure out and tell me what's happening right there. Like where would we see this, and what exactly are these parts? Okay. Well, I'm saying multiple transcripts can be made simultaneously, right? I already told you there's lots of RNA polymerase II, it's in the nucleus, you got all that. You have a lot of transcription factors, you have unique ones based on different cells, but you've got all this machinery, so as long as you have the DNA in that nucleus, all these things can be popping on and making a lot of transcripts at the same time. So it's not like you have to wait, a whole hour just to get one. In an hour, you have many, you have dozens, hundreds, thousands. So what do you think this is?
Looks like mass production of mRNA.
It's mass production of mRNA, yes. It is lots of, so you have a sequence, you have basically a TU is a transcriptional unit. Remember, it's that sequence that encodes for a gene and some other stuff in there, usually. You have a transcriptional unit going from here to here. Why do we know that? Because you've got these segments where there's nothing, right? There's nothing going on there. There's really nothing going on there, but you have these units right here where there's all this flowery fluff going on. What are those? Those are basically coming off, those are the mRNA transcripts. And as they move along, they get longer and longer and longer because the RNA polymerase is zipping along and it's giving out those tails of the mRNA that are growing. So as that processively moves along, the DNA unwinds it, building the RNA nucleotide, it gets longer and longer. So it starts short, and they get bigger, and they get bigger, and there's a lot of those enzymes hopping on, so they're all doing this simultaneously. You see that? So we have, this is an NTS, it's a non-transcribed sequence, or a non-transcribed segment. There's no transcriptional unit right there, aka there's no gene right here. The gene stuff is here, the gene stuff is here, the gene stuff is there. Okay, so this is happening, where is this happening? In a eukaryotic cell, where is this happening? Where would we?
In the nucleus.
In the nucleus. So if we're talking about a eukaryote, transcription going from DNA to mRNA is all happening inside of the nucleus. All right, so I have a video that kind of puts this together, transcription in action. I hope it plays. If not, I'll open from my original. Do I have a better one? Hold on, let me see. I have another gene. I'm just going to go forward and see if I have that actually inserted where I put everything together. If not, I'll open it on my separate little slide. Nope. Just give me a second. It doesn't want to play. Okay, so I'm going to try to open it from my other separate little folder. Courses, animations. One called beautiful animations. There we go. Okay. I'll make sure. I think the one that I put on Moodle actually plays because I embedded it. I embedded it in there. Okay. Application. Okay. hopefully it plays here. And I'll show you this file too. I think I put this PowerPoint file actually on Moodle. It's like beautiful animations. If I haven't, I'll put it on there. It goes through everything from DNA replication, transcription, to translation, to mitosis, to meiosis. It's got them all built in there in one thing. So, transcription in action. Please play.
The first step in protein synthesis is transcription of RNA from DNA. The portion of the DNA that is transcribed into an RNA molecule is called a transcription unit. An enzyme called RNA polymerase carries out transcription. With the help of protein transcription factors, it attaches to the beginning of a region of the DNA called the promoter, pries the DNA strands apart, and untwists a short portion of the double helix. RNA polymerase moves along the DNA, pairing up RNA nucleotides with their DNA complements, adding nucleotides to the end of the growing RNA molecule. Here's a close-up view of elongation. Only one DNA strand, the template strand, serves as a template for RNA synthesis. RNA polymerase moves along the DNA strand in the 3' to 5' direction, adding nucleotides to the 3' end of the RNA chain. Note that U in RNA pairs with A in DNA.
Once transcription is complete, RNA polymerase releases the completed RNA and detaches from the DNA.
So that whole section is the transcription unit. Cleaved RNA. Now mRNA can go on and be processed and chopped and sliced and diced and capped and tailed. Okay, so this PowerPoint, I think I have it on Moodle already, but I have all kinds of animations that I really like. Goes over how the leading strand is made, the lagging strand, so DNA replication overview, putting it all together, figures DNA replication, and then I showed you a lot of these videos already. Mitosis, meiosis, got all kinds of stuff. Time lapse, just to show you, meiosis again, genetic variation, a lot of different transcription. We're gonna go forward and we're gonna go to protein synthesis, we'll go to all these other ones. But these are some of my favorite animations that I like to watch myself and that I know students have used as study aids, so download that from Moodle. And I have these movies inserted throughout almost all my lecture slides, except I know this one didn't play for me. But it should play, and they should play for you. Okay, transcription. So that was that little animation, putting transcription all together. It's pretty straightforward. So that was how we make a transcript, an mRNA molecule, right? A strand, single strand. Now, remember I said that it's not, there's exons and there's introns, right? So it goes along your whole transcription unit, that whole sequence of the DNA that is a gene, or it has the gene encompassed in there, but there are exons and introns. And those exons and introns are copied by the mRNA, they're made in that message, but now they have to be taken out. And how I remember, oddly enough, the introns which you think would be kept in the mRNA, it's the opposite, they're the ones that are cut out of the RNA. So the introns are the ones that are actually clipped out and removed because they don't compose the actual gene. They're this junk in there. They're not part of the actual sequence that makes up that gene. So the introns are the ones that are cut. Okay? The exons are the ones that are spliced and put together and they remain in the final RNA. We call this RNA processing. So there's enzymes in eukaryotic nucleus that modify that pre-RNA, so we call it a pre-mRNA, before this transcript is sent out to the cytoplasm. Because remember, it's gotta be modified and capped and all these things have to be, introns have to be cut out. So during this RNA processing, both ends of the transcript are altered. So there's a cap put on and there's a tail put on. And we'll see why that's done. And then there's some interior parts that are cut out. Usually interior parts are cut out, these introns, and then the other parts are spliced together. The exons are put together, and that's why I open with that kind of fruit ninja, how we call that RNA splicing. So this RNA processing is modifying the pre-mRNA and making it the final mRNA that's going to be exported and translated into protein. So we have capping, polyadenylation of the tail, and RNA splicing. Those are the major forms of RNA processing, or post-transcriptional modification. Why? Because after you transcribe and get the mRNA, this is after you get that mRNA, it has to be modified, it has to be processed. This happens in eukaryotes, not in prokaryotes. So this is another thing that's different. There's a lot of RNA modification or post-transcriptional modifications that happen in eukaryotic cells. So what happens is there's a capping of mRNA, it makes a 5' cap. So if you imagine, you have a single-stranded RNA, and in this class, I had an application question at the very beginning, when I went over nucleic acids, and I said, which one is more stable? If you had it in a little vial in water, in an aqueous solution, you just stored it, which one would last the longest and why? Remember that? Okay, and a lot of people went, well, RNA. Well, RNA is more stable, it's shorter, it can compact up, and really the reverse is true, right? DNA is much more stable. It's huge, it's giant, it's complex around proteins, it's double-stranded, it has its partners wound down, it lasts for much longer. RNA is more fleeting and degrades much more quickly. It's single-stranded, tends to be shorter. So this is a way that eukaryotic cells go, you know what, let's try to prevent it from degradation, from too many enzymes chomping it away. Let's at least let it be alive for a little while so that we can turn these into the protein that they're supposed to be. It caps them with this methylguanosine. It's basically a little methyl group on guanosine, okay, so like the base. It makes a little 7-methylguanosine cap that adds on to the 5' section of that mRNA transcript, okay, so it's capped. And it basically is a cap on the 5' end, situated though in the wrong direction, so look, it's kind of upside down. So it makes it a little more resistant to degradation or any enzymes binding on there, so it's in the reverse direction. Methylguanosine cap that's added, oops, on the 5' end. So here's the 5' end of the RNA, flipped upside down, here's the sugar, here's the phosphate, here's the guanosine. Okay, so it's a little cap. We also add, eukaryotic cells add a tail, an adenylation, a poly-A tail to the end. So multiple adenosine monophosphates are added to the 3' end. We call that polyadenylation. So adenosine, monophosphate, added a whole section of them, boom, 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 boom. Enzymes add a tail onto it. 20 to 200 adenines are added to the end of the mRNA transcript. This is the post-transcriptional modifications. Stretch of RNA that has only adenine bases, so it's all A at the end. That's the poly-A tail. So what it ends up looking like, you have a little 5' methyl, 7-methylguanosine cap in the little reverse direction on the 5' end. You have a poly-A tail anywhere from 20 to 100 to 200. A bunch of adenosines out at the end. Why is mRNA modified? Why do we have this crazy cap, this guanosine cap, and this poly-A tail, and then ultimately what I'm going to show you right now is they get fruit ninja'd up and all the introns get spliced out. Why do all of this to RNA? Linking it back to that first application question that I asked you about DNA RNA. Why do all of this to RNA? You don't want RNA to be degraded immediately, right? It's to add to the stability and the longevity of RNA so that it can safely leave the nucleus, go out into the cytoplasm and not be broken down and fall apart. This is your message, this is your transcript carrying the genetic information out to the cell so it can make a protein from it. So you really have to make sure it's stable and it at least stays around a little bit so that you can get protein from it. So it helps to facilitate the export of the mRNA, capping it, tailing it, all that, splicing it. Helps to facilitate, protects the RNA from hydrolytic enzymes. That means enzymes that use water and hydrolysis to cut, break things apart. Remember, this is a polymer. RNA is a polymer, and it can get hydrolyzed or broken down. Remember that hydrolysis reaction that uses hydrolytic enzymes? That can happen. It also increases the longevity or the lifespan of that mRNA, or those mRNAs, so they can last longer. It also helps ribosomes attach to the 5' end for translation. So for turning that transcript into a polypeptide. So helping in the translation, facilitating its export out of the nucleus, and keeping it alive and around for longer so that your cells can actually go, okay, factory workers, now let's make protein from this transcript that we just made. Yes? And we're going to talk about translation. Next time, I'll end here, we'll go over splicing, and I'll wrap it up and go into translation. So make sure you review that section of the book and the exercise on translation and protein culture. I have application questions in here too at the end. Please do those. You can bring them on Friday. It's related to this exact material, and then we'll go over it in class again.
Do you want me to end this for you?
Oh yeah, you can stop. Thank you.